Hope y'all enjoyed this part of your evening. I turned my collars on.
Great, thanks very much. We're going to get started. Our first speaker of the new year in the Broad MPG Primer series is Heidi Rehm. Heidi's going to be speaking on the subject of clinical interpretation of sequence variants in genes. Heidi is the director of the Partners Healthcare Laboratory for Molecular Medicine, medical director for the Broad Institute Clinical Research Sequencing Program, and an associate professor of pathology at Brigham and Women's and Harvard Medical School. We're very lucky to have Heidi speaking today. Thanks.
Great. So thank you, everyone, for coming to learn about clinical variant interpretation. I did add one minor addition, which is and genes. So you really can't effectively interpret variants if you don't use some effort to interpret the gene-disease relationship to start with. And we're actually going to start with variant interpretation, but then I'm going to switch halfway through and actually talk about gene-disease association and assessing the validity of those associations. So one thing that I think is really important to keep in mind is when we're thinking about clinical variant interpretation, for the majority of what is tested clinically for patients today, it's really around monogenic diseases, not complex traits and GWAS hits, which has very little clinical utility in today. So most of what we do is we interpret variants in genes associated with monogenic disease or Mendelian genes. And the experience from my lab in the first 15,000 probands that we have tested for monogenic disease testing showed that the vast majority of variants are extraordinarily rare. You know, the variants that we are able to interpret and report as potentially or known of clinical relevance, most of them are seen once and we never see them again. You get one shot at interpreting that variant. There's no publications, nothing out there, and we will never see it again. In a lab of my size, you know, over 15,000 probands being tested. So it's an incredibly challenging activity because you can't just wait till somebody else figures it out. You really have to think about what can you figure out on a variant with one case in hand. Now, in other cases, sometimes we do see variants over and over again, and you can build evidence over time. Although sometimes those variants that you see over and over again, you still can't interpret them. So it's a challenge. The other challenge that's out there is that we don't interpret variants consistently. And a paper we published in 2015, after a number of labs had started submitting to ClinVar, we were able to show that at that point in time, 11% of variants that had more than one interpretation so we could compare, 17% of those variants were interpreted differently. So there's a lack of consistency in how this is done. And I can tell you that the first lawsuit of a patient who claimed the child's death was due to a lab's misinterpretation of the variant has already happened. It happened last year when Quest's Athena lab was sued. That lawsuit is still underway. So misinterpretation of variants can have dramatic impacts on patients' lives. So we have to figure out how to do this effectively. We have been tracking discrepancies in ClinVar. We've actually now moved from 17% down to 15% discrepancies, and we hope that over time we will continue to improve that, largely through the process of what I call share and compare, identification of discrepancies and resolution of those discrepancies. Now, one of the reasons that we were fairly discrepant is there was no standard for how you interpret evidence for a variant for a long time. We recently published a standard. This is the group of us that put this standard together after 2 years of effort. And, you know, drinking all of the wine, that was because we largely couldn't agree on much of anything without a little facilitation. So, you know, this still is largely a professional opinion process. You know, do I think this evidence is valid or not? We did do our best to try to make it more objective, but it's hard. We spent, 2 years, weekly conference calls. We sent surveys to labs. We held open forums with the community, all sorts of things to try to arrive at, this is one survey just trying to ask the question of the appropriate terminology Labs would call things deleterious, associated, mutation, polymorphism, normal, not associated, non-pathogenic. They use terms like possibly, probably, likely. It's like we can't even compare if we don't use the same term. So we put out a standard of 5 terms for Mendelian disease variant description of pathogenicity: pathogenic, likely pathogenic, uncertain significance, likely benign, and benign. And now largely the entire field has adopted these in the last year. One other thing that I think is really critical and a lot of people mix up is the process of variant classification versus the interpretation of cause of disease in an individual. It's 2 separate steps. So, first we ask, does this variant actually impact the gene or protein with some, impact to function? But then the second question is, can that impact lead to a diseased phenotype? Is it, when we talk about pathogenicity, that's really with respect to a disease. Is this variant pathogenic for cystic fibrosis? Is it pathogenic for breast cancer? You can have a variant that is a null and disrupts protein function, yet has no effect on phenotype, right? Maybe because the gene is duplicated and loss of one doesn't do anything, or maybe there's just no disease phenotype by losing the function. There are some genes where you only get a phenotype if you have a gain of function, and loss of function has no impact. So, these are things that we have to think about, is what that impact and does it cause a disease phenotype. But separately, when I have a patient in front of me, I can find a pathogenic variant in one gene and report that out as pathogenic, but then say it has nothing to do with the indication for testing that patient showed up, because I don't think that variant is actually explaining their disease. So there's 2 separate, especially when we do clinical lab reporting, we first evaluate variants, and then we say, are any of these variants actually causing the disease the patient was referred for? And those may or may not be the same answer. So with that terminology, we use the term likely. Now that's a bit non-quantitative. There is no formal definition in the Webster Dictionary or Oxford Dictionary of what the word likely means. But we found that it was useful to be able to say, look, we're not sure about this, but we feel pretty good. But we felt the need to define that quantitatively for the field. And so by virtue of a vote, we defined 90% to be the definition of likely in the context of clinical variant interpretation. Not because there's a science there, but because we felt that clinicians would value the report of a variant that we felt roughly 90% confident in its interpretation. Much of medicine is based on guesses. I think you broke your arm, and so therefore I think you should do this. I think you have a torn ligament. I think. Those are just guesses in a lot of cases, but it's still useful in medicine to make educated guesses, and so this is sort of the equivalent of an educated guess. Now, what we did was we came up with a series of pieces of evidence that in this list would support pathogenicity calls. And then we have a separate list of evidence that would support a benign call. And going backwards here, we then rank them in terms of how strong, is it very strong, strong, moderate, or supporting, each of those pieces of evidence were. So for example, if you had a predicted truncating variant or predicted null variant in a gene where loss of function is a known mechanism, that's a very strong piece of evidence. If it was the same amino acid change as a previously established pathogenic variant, so it was a different nucleotide change, but had the same effect in terms of changing the protein, that would be a fairly strong piece of evidence. If it was a de novo variant in a patient with de novo disease, in a gene already well established as cause of disease, that would be strong evidence. There is well-established in vitro or in vivo functional studies showing impact. Now, as I go down this list, I should clarify that this is the short version. If you get our guideline, which is like 15 pages long, there's a lot of detail behind each of these statements as to how to apply these rules that are very, very important. For instance, when you observe a de novo variant in a whole exome study, and you find it in a gene that's not been well studied, you cannot assume that is pathogenic, because we all have a de novo variant in our coding region across, if I sequence any of you and your parents and do a trio analysis. However, if that de novo variant is in a gene it's already well established as a cause of the patient's phenotype, then your a priori likelihood of a de novo variant showing up by random chance in that gene is much lower, and you can assume that that's strong evidence. Prevalence of variant in cases is statistically significantly increased over controls, strong evidence. Moderate, located in a hotspot or critical well-established functional domain, absent from controls. This is a little outdated, this particular slide. Now we use exact nomad. For recessive disorders detected in trans with a well-established pathogenic variant. In-frame deletions or insertions in a non-repeat region or a stop-loss variant. Novel missense changes where a different missense change at that same site is pathogenic. Assumed de novo, but you haven't confirmed maternity and paternity. This unfortunately happens a fair bit. We find what appears to be de novo variant and then find out dad is not actually dad. So that piece of evidence is critical. Co-segregation with disease in multiple affected families. We're going to talk a little bit more about segregation and the quantification of it later. Missense variant in a gene that has a low rate of benign missense variation. These, none of these are really strong pieces of evidence down here, but they kind of provide some support. Patient's phenotype is highly specific for a disease with a single genetic etiology. Multiple lines of computation, although these are things like polyphen, CAD, et cetera, that support. Is almost never relevant, but we had this situation where Myriad wasn't sharing its data, but we captured a bunch of it through a project, and we could point to Myriad's experience in breast cancer testing, but they weren't willing to provide their evidence, so we used that as a piece of supporting evidence if they claimed it was pathogenic or benign. On the benign side, and again, this guidance is for Mendelian disease classification only, so this obviously wouldn't work for complex traits, but for Mendelian disease, the allele frequency is above 5%. You can pretty much rule it out for all Mendelian diseases, given the frequency issue, as long as there isn't any published contradictory evidence. We're actually in the process of defining any alleles above 5% that are actually disease-causing. Most of them are low-penetrant alleles, but this is quite rare. And then, and we're actually, most of these are sort of the opposite of the pathogenic ones, like well-established functional studies show no effect, lack of segregation, et cetera. So I'm going to go through some variants to actually give you examples of classification. So that same list of evidence is also, I put it on a grid with pretty colors, showing the strength increasing for pathogenicity, the different types of data, population, computational and predictive, functional, segregation, de novo, allelic data, other databases, and other stuff. And then there's a set of rules that allow you to compare, combine different pieces of evidence to form a pathogenic call, a likely pathogenic. So you get one of these and two of those on all the possible combinations to arrive at one of these. So, this is not the only method to do variant classification, but we developed it as sort of the default method if you don't have a better one for a particular gene. There are some groups who have come up with specific methods for specific genes, like the INSIGHT program for colon cancer has a specific methodology more akin to the types of data that you see on mismatch repair genes, for example. Enigma has come up with an approach for classification of variants in breast cancer genes based on the types of data you see from families with breast cancer. So there are other ways to do it, some of them probably more statistically valid, in fact. But you have to have specific data types to come up with these more specific methods. So we sort of put this out as a default method for any gene that doesn't have a more specific approach. Now, I'm going to give you an example of one variant. We're going to walk through the classification, and I'll also highlight some of the challenges. Just because we have a standardized approach, it still requires a fair bit of judgment and opinion, and therefore people will still arrive at different answers. And in fact, we ask people to use the ACMG guideline and classify one particular variant. This is a variant in FBN1, which is the gene for Marfan syndrome. And these were the set of conclusions we got from the 6 different labs. 2 people in one lab evaluated, one said pathogenic, one said likely path. Another lab, likely path. Another lab, 2 people. One said likely path, one said VOS. 3 other labs said VOS. So even with the same rules, you still arrive at different approaches. So now, anytime you evaluate a variant, you need to understand basic things about that disease and that gene. So in the context for this particular variant, the primary disease this gene is associated with is Marfan syndrome. It has skeletal, ocular, and cardiovascular phenotypes. The known mutation spectrum for this particular gene associated with Marfan syndrome is pretty much all types can cause disease. Missense, inframe tend to be more severe than null variants in this case, but both null variants and missense and inframe are all shown to be pathogenic, or specific ones. There's extensive intrafamilial and interfamilial clinical variability, so different patients can present with different degrees of the phenotype, but the penetrance is quite high. And so nearly all Marfan syndrome patients have an affected parent or the variant is de novo. That's not true of all Mendelian disease, but in this case, it's fairly consistent. There are other phenotypes that have been described with varying levels of evidence. Why is this important? Because when you read papers to try to get evidence, if they said we found this in a proband, well, if the proband doesn't have the disease, that's not good evidence to use. You really need to understand the phenotype to make sure that you're appropriately evaluating the cases in the literature. So why did all these groups come up with different answers? So first, you can look at case data and control data. So each of the labs went out looking for this data in the literature. Lab 1 found 11 cases out of a total of 2946 compared, and at this time we had access to ESP. This was before EXAC, this particular case that we did, and they actually calculated an odds ratio based on this data. Lab 2 only found 3 cases in the literature, so this is a challenge right here. Trying to find these cases in the literature and search on the variant is actually not straightforward. We actually use a tool that generates a search string that gives all the possible cDNA nomenclatures, protein nomenclatures, 3 amino acid, single amino acid, all these different options, different transcripts have different nomenclature to try to come up with the cases. And that's not trivial. So not surprisingly, all these groups came up with different numbers of cases when they went to the literature. This group said it's either 7 or 9, and it depends on whether that second paper I read actually re-reports the cases in the first paper. Not quite sure. That happens all the time. You publish your first study, and you publish a larger study that actually included the cases that you first published. And we often only figure this out by looking at overlapping authorship. This is a huge challenge. One day, a student came to me and said, this variant's been reported 5 times. And I went back and looked, and it turns out it was the same patient in all 5 papers. And in fact, the patient we were testing in the lab was the subject in all 5 papers, and he had never gotten his results back. There was one patient in the world with this mutation. So this is a challenge that we deal with quite a lot. Also, coming up with denominators is not straightforward sometimes, especially when you're using databases like ClinVar. There's often no denominator. A lab will submit a variant, and sometimes they'll say how many cases they saw it in. You have no idea out of what number of cases they've tested. So sometimes people will make up numbers like, well, I got 2,946 because that was the total cases reported in the locus-specific database. Well, that's not really a good way to do it. So others will, just aggregate from the literature. Each paper often says, we tested 100 patients, we tested, 90 patients, et cetera. You add them up. So these are all the challenges with trying to do case control comparisons, and often you're comparing to exact ESP, groups that haven't been well phenotyped. Sometimes you're comparing to people that have. I also pointed out the varying clinical phenotypes. So if you actually look at all these cases, they didn't all have Marfan syndrome. Some had Marfanoid-like features. Some had Sprinson-Goldberg, which has to date now been ruled out as an association with Marfan. So it's not even clear that you should have added up all of these cases specifically in this example. And that's where really understanding the phenotype, you're going through literature, is critical. So at the end of the day, if you use the ACMG guideline, one can say, well, some of these, depending on which number you use, there's probably a statistical difference. But you could drop the evidence to moderate because it really has a number of weaknesses in the data. That's probably the best that I would give this data in this case. And some judgment is going to be used to make that assessment. Now, in this second line of evidence types, it's sort of computational predictions, and each of the labs used a series of different computational prediction algorithms. You can see by the darker squares which ones they use. You can see they all use different ones. However, pretty much all the labs said they all got different answers across all of these and didn't bother even using any of this evidence. Now, one of these pieces of evidence, a novel missense change at an amino acid residue where a different pathogenic missense change has been seen before. So one group said, well, actually, the variant is at the same site as another variant proline to serine, and therefore they use that as a piece of evidence. However, you can only use that if that variant actually has evidence for pathogenicity. When we looked at it, there was one case reported with this variant, no other data, no segregation, nothing. So we said, no, that's not pathogenic. You can't use that to support your other variant, sort of a circular argument. So basically, there's no evidence here. The computational data is unhelpful, and you can't apply this rule. This rule is often misused. Locate a mutational hotspot or known functional domain. So you can go. Do you have a question?
So, yeah, so going back to that last point about using a previously reported variant.
Yep.
So do you find yourself that you have to apply the criteria that you've developed, SCGME, retrospectively,
yes,
every time you invoke pathogenicity of a previous variant? So
yep.
it's almost like a...
Yeah, you kind of keep going back.
Right. Repeat the question.
So, sorry, the question was around, when you're comparing it to another variant for this particular rule, do you have to go then evaluate that prior variant with ACMG criteria? And the answer is yes. You have to evaluate that, make sure it meets pathogenicity criteria before you can then decide to apply this rule. So yes. Okay, so in terms of functional domain, this is not like you go into UniProt and there's some line that says this is an IG domain. There has to be a lot of data saying that this very discrete element is a well-established functional domain where all pathogenic variation leads to disease and there is no benign variation in the region. Now, the size of that region could be variable. It could be, 2 amino acids. It could be 10 amino acids. An example might be the pore domain of some of the channel proteins, where pretty much any variant in a stretch of 10 amino acids might be viewed as fairly disruptive. The glycine residues in the collagen protein. So there's certain functional domains that, the critical cleavage, amino acid for a particular enzyme. So you really have to know that functional domain, really feel it's devoid and intolerant to any pathogenic variation before you can apply this. Or it's a specific hotspot that repeatedly gets mutated. So most of the labs did not consider this evidence by just looking up in UniProt and showing it was calcium binding region. One group did actually call it moderate evidence, which we all voted was not correct usage of this rule. Here's an example. There's actually not a lot of proteins that have, such a discrete, all the pathogenic variation is littered here. There's no benign variation, and all this region is only benign variants. So some proteins have some of these regional things, but a lot of them don't, and you often can't use this rule. Another part of the rule that's not relevant here because there was no functional data, but the functional rule where a well-validated assay shows a functional effect. A good example of that would be particularly for enzymes. So when we find variants of uncertain significance in the GLA gene, which is the alpha-galactosidase enzyme for Fabry disease, the assay that is used is actually an enzyme activity assay that can be run on the patient's blood and show an actual activity readout in each patient so that you can use that to specifically assess whether the variant has a deleterious effect on function. That is very good evidence. In contrast, a lot of functional assays are not well validated. this question of non-segregation with disease or segregation. So I should clarify here, one of the very big battles I had with the working group was I wanted to quantify segregation. Obviously, it's one of the most quantifiable pieces of evidence, and I was voted down. So the actual guideline ended up with this co-segregation with disease and multiple affected family members as a supporting piece of evidence that you can increase using your own judgment, because we couldn't, in the committee, actually agree on how to quantify and give guidance. So ClinGen, we are working on giving specific quantitative guidance. I'll show on a slide in a second. But the guideline actually, unfortunately, does not give you that specific guidance. So you have to use your judgment until we come out with the better guidance.
What are the arguments against quantitation? I can't imagine.
Could you repeat the question?
The question was, what are the arguments against quantitation? So one of the arguments, which is a valid argument, is that when you use segregation, you are implicating a locus, not the specific variant, right? And that is a very valid argument. And so that said, when you have a variant in a well-established gene associated to that phenotype, and you've sequenced the whole gene, you can often be pretty sure that that is the variant that's relevant. Not 100%, but I would argue a log score of 3 for a gene already, or a variant in a gene already implicated in disease is actually incredibly strong evidence. We use it all the time in cardiomyopathy, particularly diseases where all the variants are in the coding region. And if you've sequenced the whole coding region, you can pretty much rule out that, or assume you haven't missed anything. So that was one argument, was that this question of, is it in linkage to disequilibrium versus the cause of variant? The other argument was a less intelligent one, which was clinical lab directors don't know how to calculate log scores. I said, I can provide them a little table. They don't have to calculate it. So, which I did. But anyway, so I'll stop bitching about that. We're just going to solve it after the fact. So now the question of non-segregation with disease. So in this case, this particular variant, so one lab had clinical testing data from a family. This was a 3-year-old with Marfan syndrome. And the father had tested positive for the genotype, but was phenotypically negative. So there's different types of non-segregation data. So one type, maybe I have a slide here. So if we assume all the pink or red, whatever color this is, are affected, then for all the people affected, you want to see segregation of the variant with the disease, right? And the more you see, the higher the log score, the more statistically significant. And so if this person was affected, but the variant came from this side, well, that would be non-segregation. And that would be very strong evidence against this variant being pathogenic or causative for disease, at least in this family, unless there was a risk of a phenocopy. So for certain phenotypes, a patient can be affected, but actually due to something entirely unrelated to the hereditary disease. So you have to always be careful that the patient, each family member has been well diagnosed, and be wary of phenotypes like breast cancer, for example. A woman can have breast cancer due to a hereditary cause, but 1 in 8 women have breast cancer. So if you happen to have a person in a pedigree who had breast cancer, but the variant didn't segregate, you could realistically argue, well, maybe that woman had breast cancer for a non-hereditary genetic reason. On the other hand, a very concrete diagnosis of Marfan syndrome with all the full features, you could pretty much assume did not arise due to some environmental cause. So you have to be wary of your phenotyping. But if you show non-segregation, you're absolutely sure of the phenotype, then that's pretty strong evidence against the variant being pathogenic. In contrast, this particular evidence where the father is genotype positive but didn't develop the phenotype, well, a lot of disorders, particularly dominant ones, are not fully penetrant. So genotype positive, phenotype negative, we often just don't even count unless the disease is 100% fully penetrant, because that's just, caveated evidence. So, in that case, I think I said, well, you could count that as supporting evidence, in part because Marfan syndrome, as I told you earlier on, is actually fairly highly penetrant. So because of this, I wouldn't count it as strong evidence, but I might count it as supporting evidence, sort of shift the strength over. Now, I told you that ClinGen is working on providing quantitative guidance around segregation data. We haven't yet arrived at a final proposal. There is actually a publication from Gail Jarvik's group that's been out there, and they actually used the grid we had in the ACMG guideline and then filled in quantitative data, where they proposed that in a single family, if you have 3 segregations, that would count as supporting evidence, 4 as moderate, and 5 or more as strong. And that was Gail's proposal. If you were using data across multiple families, that you drop that by 1 to 2, 3, and 4 or more. We have 2 of our expert panels, one in cardiomyopathy, one in RASopathies, propose slightly different numbers. So we're we have a sequence variant interpretation work group that is deliberating over what should be the formal guidance as to how many segregations should count as what. It's still a bit hand-waving. This is not a highly quantitative approach. It is largely professional opinion, but at least there is some quantitation here. So that's where we are right now. I talked about de novo data. So we use a de novo occurrence, again, in a gene that's already associated with disease, for which you have confirmed the paternity-maternity relationships. You know it's de novo. That's strong evidence versus moderate. In this particular variant, there was no de novo data, so that didn't get applied. I did want to point out that, and I sort of mentioned this earlier, if you have a de novo event in a gene without a known associated phenotype, that is a VUS in a candidate gene until other evidence is generated to implicate that gene. Again, for the very fact that we all have, every one of us, a de novo variant somewhere in our coding region. And so that is not a statistical rare thing if you look across the genome. On the other hand, if you are looking at a gene that is already implicated in the patient's phenotype, that changes the statistics, and you can use that as stronger evidence. Okay, in this particular case, there was no allelic data, but the types of allelic data, so when we do recessive disease testing, if you've got a patient with a phenotype, let's say hearing loss, often recessive, and you find, or let's say Usher syndrome, hearing loss and blindness, and you find a pathogenic variant in the USH2A gene, a gene already known to cause Usher syndrome, in one allele, and then you find a novel missense variant in the other allele, the sort of guilty by association phenomenon provides some support for that variant in the other allele because you've sort of already implicated that gene at some level by that first variant. Now, that's not a guarantee because we can be carriers for recessive pathogenic alleles that have nothing to do with what disease the patient has. So it's not definitive evidence, but it is, we considered a moderate piece of evidence to find it. Now, if you have 10 families, 10 pathogenic variants, and this allele is always showing up as a second hit, the more observations, the stronger the data. So you can kind of move this up in strength with multiple observations. On the other hand, the data that would exclude or provide supporting evidence against pathogenicity is if you observe the variant in trans with a dominant variant. For example, a lot of the way that Enigma uses data for breast cancer variant interpretation is if they have a known pathogenic variant in one allele, and then a second variant is found on the other allele, and the patient does not have Fanconi anemia, which is due to biallelic BRCA1 and 2 variants, they can assume that that second variant on the other allele must be benign. This is also how the CF community has done cystic fibrosis variant interpretation. You've got Delta 508 on one allele. Any variant found on the other allele has to be benign in a father who has given rise to children because he is infertile if the second variant is pathogenic. So they've used that as a way to sort of rule out variants by this sort of trans observation. It's very useful. We also often would say a variant insists with a pathogenic variant is probably supporting evidence for a benign role. It's usually not relevant, although you can't completely exclude it. So this is an example of a case in the lab. We found one pathogenic variant in the GJB2 gene in a kid with hearing loss, but then there was a VUS on the other allele. How do we interpret that? Well, if you test parents and you find that that variant is not on the other allele, it's actually in cis, we would often interpret that as likely benign. In contrast, if you were able to show the variant was on the other allele, that remains a VUS, but it provides some supporting evidence towards pathogenicity. Okay, so we also have a rule, patient's phenotype or family history is highly specific for the gene. This really comes down to the extent of association of a unique set of phenotypic features that are highly specific for a certain gene. And the more specific that association, the more that evidence is that any variant you might find in that gene is more likely to be pathogenic. It's not a strong piece of evidence, but the more specific that phenotype is for that particular gene, you can sometimes use that as a supporting piece of evidence. I see this misused all the time. Like, patient has autism, and this is an autism gene, therefore that's a strong association. No, that's not a specific association for pretty much any gene for autism. It's a fairly common phenotype, and there is not like a single gene that causes autism. So that's an example where you would not want to use that, but there are some highly specific syndromes with, pretty much if you're diagnosed with that syndrome, you will have a mutation in that gene. So in this case, this would be, actually I was arguing because all of the patients that were described in the literature actually have quite different phenotypes. I was arguing that's argument against pathogenicity. Usually with the same variant, you're more likely to see the same phenotype. And we often use this in cardiomyopathy evaluation, where the MYH7 gene can either cause hypertrophic cardiomyopathy or dilated cardiomyopathy, but they're different mutations that cause each. So if we test a cohort and we find a variant and half the patients have HCM and half the patients have DCM, more likely to conclude that that's probably a benign variant. It does not cause either disease because of the variety of phenotypes. So with all of that sort of assessment, I somewhat probably too generous gave the case control data a moderate, piece of evidence. But the other pieces that we concluded were on the benign side. So for sure, at this point, this is a VUS at best, based on the data that was provided. Yes. Is there a question? Yes.
Just curious, in the panel's discussion, was there ever any consideration to the idea of separating genotype and phenotype, given the idea that the patient phenotype often, as reported to the clinical lab, is maybe incorrect or maybe incomplete, or they may be actually leading the witness? Just curious your thoughts and the committee's thoughts over the history of this.
Yeah, so the question is, have we considered just simply separating out the question of the phenotype from the evaluation of evidence? Because it is not easy sometimes to actually have in hand an accurate phenotype, either in the clinical lab in terms of what you're provided, nor frankly in some of the data you try to find in the literature. So the answer is no, in the sense that while that is not perfect data, it is better than no data. Like if we had gone through this exercise and just added up all those cases and just said, we're not going to consider the phenotype because it's not great, then I would have been more wrong in this case, right? So while it is not perfect data, and I can tell you that in the clinical lab, we often don't get phenotype, but when we have a variant we don't understand, we call back the physician and say, we cannot interpret this variant until you provide us some more phenotype. And I can say that 100% of the time the physician then takes the time to give me phenotype, because now the variant interpretation is on the line. So it does require follow-up. I often email authors and papers all the time. You reported this variant, can you provide me with more detail about the phenotype? I do that all the time, our fellows do that all the time. So the answer is yes, this is challenging, but it will help you if you can get that data. So.
That's a really important point. I totally agree. I wonder it might be useful to also consider a score that you give before you consider the patient phenotype, and then a score you give after, because you're absolutely right. Yeah, obviously, you're absolutely right. You need to consider it for the clinical assessment of that patient. But you could also kind of make a more consistent score if you maybe perhaps blinded to that. Just throwing that out.
Yeah, I mean, it's possible that then you double people's effort. They kind of go through the process, and do they just discount all that case data because they don't have phenotype? Like so many of these pieces of evidence kind of rely on the phenotype. Segregation. You can't do segregation if you don't know the patient's phenotypes. You can't do case control. You can't assess the specificity of phenotype for the genotype. So there's so many of these cases, de novo occurrence. We've got to know if the parents were affected or unaffected. So many of the rules do require you to understand the phenotypic presentation of the proband and or the family members that I think it would be really just annoying to try to do it without and then do it again with. You basically have to make a judgment each time you're looking at a set of evidence. And the more phenotypic data, usually the better you can judge. But it's not such a concrete, well, here's the process without phenotype and here's the process with. Most of the data, half of it, you just can't use if you don't have phenotype. So,
what is the process by which these exchanges that you have with individual authors or clinicians actually update the literature? So after it has been published, you gather all this information from a lot of hard work.
Yeah.
How do you prevent other people coming to do the same thing in the future?
I can tell you exactly how we do it.
Can you repeat the question?
Yes. So the question is, I have all these conversations with people around the world to help us interpret variants. Does that ever get into the literature? The answer is largely no. However, I can tell you where it does get is that when I write a clinical report, we summarize all of the evidence that we have found on the variant. And we not only put that in the clinical report, but we store it and we constantly update our variant interpretations in our database, and that is what we submit to ClinVar. So if you go into ClinVar and you type in the variant, and you come up with a lab listing, and then you click on summary evidence, you'll get to another tab. And if you go, you'll see a text box, and maybe about roughly half of the labs, GeneDx and Vitae, my lab, a lot of us submit a full summary of all of the evidence. That will point to this exact personal communications, other lab data, all this stuff we write in summary there. It's not highly structured, but it at least is provided, and that is how it's getting out there. I've watched other clinical labs excerpt stuff from my stuff in ClinVar and put it in their clinical report. So it's helping. It's not the most computationally supported process, but it is getting out there. So I did want to revisit this question because I see people misinterpret truncating variants all of the time. They just assume truncating equals pathogenic. No. So there's a key piece here, only when it's a known mechanism of disease. You have to make sure you've looked at the gene level. Get into that in a minute. There's also other critical things, and we've actually come up with our own predicted null checklist every time we find a predicted null variant as to what you need to check. So I already asked the first question, are null variants an established mechanism of disease? Can't call a variant pathogenic if you haven't assessed that and concluded yes. How frequently are predicted null variants found in the gene in large population databases? This is actually when the constraint scores from Exact and now Nomad are incredibly useful. So you can look up the constraint score, and if it's high, it says there's pressure against null variants in that gene. Now, the scores as they're calculated today are not good for recessive genes, although my understanding is Caitlin is working on that, the team. So hopefully we'll have better recessive constraint scores. So, are there biallelic observations of loss of function when you're dealing with a recessive gene? Sometimes you need to look at the specific exon level. Could be lots of loss of function variants in particular exon, but that exon is alternatively spliced and not a critical part of the gene. So you sometimes have to get into the exon level assessment. And more specifically, is this exon alternatively spliced so that your predicted loss of function variant is in an exon? It doesn't matter. That's really important to interpret a variant. One useful thing is, are there any other known pathogenic variants in that exon? It doesn't have to be a loss of function. Any known, well-established pathogenic variant says that that exon is critical. and then you can assume that other variants in the same exon would be also impactful. Is nonsense-mediated decay predicted? So while the vast majority, though not all, genes will undergo what's called nonsense-mediated decay, and basically what that means is the stability of the transcript requires ribosomes to be across the entirety of it up to a point in time, and if they are devoid of ribosomes because of a nonsense or a frameshift variant that's upstream, it will rapidly degrade the transcript and you won't get any protein. So a lot of these upstream predicted null variants just simply lead to loss of the transcript. And you can largely assume that if it is upstream of 50 nucleotides upstream from the last exon-intron or exon-exon junction. So what it means is you can't assume nonsense-mediated decay if it's in the last exon. If it's in the penultimate exon and it's upstream of the last 50 nucleotides, and most transcripts will degrade. So that's sort of the boundary. Now, I should mention that 50 base pair stretch has to be where the newly formed stop codon is of a frameshift, not actually where the variant is if it's a frameshift. It's because if you have a frameshift, the reading frame changes, and then a few more amino acids can be added. And if that extends into the 50 base pairs, then it won't undergo nonsense-mediated decay. So it's where that new stop codon is predicted. So it's not a perfect process, but you can largely assume things upstream of that will lead to nonsense-mediated decay. And sometimes it's just simply useful to know, are there any predicted null variants downstream of the one that you've seen? If things downstream cause disease, then probably things upstream will cause disease. Sometimes if you're dealing with the very beginning of the protein, you can have alternative starts. And so are there variants upstream of your variant too? And here's actually a case that we just had in our BabySeq study, and Tim is well versed with this case because we had a lot of debate. We found this variant, 44+1, canonical splice site. Most people assume +1 and +2 variants are definitely pathogenic, but if that exon is alternatively transcribed, then it may have no impact. And this variant is in the first exon. This was the main transcript reported in throughout the literature that included this exon 1, but there were no pathogenic variants ever reported in exon 1. So we had a little skepticism. We actually looked at found the GTEx data, and oops, I'm going to skip ahead, which showed that almost none of the tissues included that upstream exon. Only testis had it. So we were like, maybe this isn't pathogenic, and this variant has no effect on most of the useful transcript. However, in this particular case, we then went and checked the newborn screening results, and the baby actually failed their biotinidase deficiency newborn screen. Then it was repeated, and they passed the second time, and the baby was given a clean bill of health. We then recontacted them. A physician ran an enzyme level assessment showing it was decreased, and they had partial biotinidase deficiency, and that baby was now put on biotin supplementation. So the point is, it's tricky. So it's possible that this data is not representative of temporal changes in expression and other things. So the bottom line is you need to go in and look at transcripts and find out, and in this case, exon 1 was present in 3 out of 5 and absent in 2 out of 5. So you can't just assume that this will have an impact, and you probably need other evidence to prove that. In this case, we had to use clinical data. Okay, so the whole process of adding up all these evidence codes using the ACMG guideline process is actually quite annoying. And so one of the things that we've done in ClinGen is we've developed a curation interface, and I don't have time, or actually not even going to have time for much of the gene evidence stuff, but there is a tool that has been launched. We're currently stress testing it within ClinGen, but it will be released to the community soon, where it sort of guides you through all of the questions of the ACMG guideline. You can say that things are met or not met. It actually tracks all the codes up here. If it's colored, it's met. If it's gray, it's not met. If it's not evaluated, it's just white. when you're done, it gives you a summary of all the things you've said have been met or unmet. It allows you to modify the strength of something. I showed you examples where I took the evidence but said, but it's a little weaker than it should be or stronger. And then in the end, you can create your interpretation. You can override what the system gives you using your professional judgment, but it gives you like what the calculation is. And then you assign it to a disease and an inheritance. And so it sort of adds everything up. It even, as you're going along, gives you a summary of where you are. You've got no benign, 3 strongs, 1 moderate, and right now it's giving you a pathogenic score. So you can sort of see, it'll constantly update. So that will soon be released. You can actually anybody can look at the test demo right now and try it out, although your data will not be permanently saved. And we'll soon be giving people access to the production instance to actually use it to curate variants. So that's coming. So we have like 5 minutes to go through gene level evidence. I love this slide that Daniel put together. So we have known pathogenic variants in known disease genes, Delta 508 and CFTR. Nobody would dispute that. We also have variants of uncertain significance in genes that we well understand today, a VUS and CFTR. And if we certainly have variants we don't understand in genes we don't understand, the VUS and the goose. This cannot exist. You cannot have a pathogenic variant in a gene for which you don't know its disease association, right? You can say that there's a null variant that might disrupt the protein, but you can't say it's pathogenic for disease if you've never implicated the gene in disease. So it's really important to establish the gene level strength of evidence before you go through the process of evaluating a variant. And we're not going to have time to do this, but I'm going to very quickly just breeze through, and maybe we can have another primer at some point in the future and do gene level evidence. But ClinGen has come up with a series of strengths of gene disease validity: definitive, strong, moderate, limited, no evidence, conflicting, either disputed or refuted. It's on our website. I'm going to skip through some of this and just get to some interesting cases here, but we just don't have time for it. Clearly, I was more ambitious. So we go through the process of assessing genetic evidence, case-level data, family segregation data, case-control data, burden effect, various statistical analyses, as well as experimental data, different functional type assessments, animal models, these sorts of things. And so this is like the genetic evidence scoring. It's a dominant disease proposed, and you have a proband with a non-loss of function but shows some impact, or a loss of function, or a de novo variant, or for recessive, 2 in trans, 1 is LOF or de novo, using LOD score data. So there's all sorts of points that you assign for every proband with a different type of data. If you have case control data, once you evaluate a lot of parameters around statistical significance, you can assign certain points. So that's sort of how we score genetic evidence. I'm going to skip ahead here for a second. And then there's functional evidence, and of different types. And this actually follows the MacArthur paper that we published after a workshop a few years ago, published here. And it looks at biochemical function, interaction data, expression data, model systems, and then scores based on different types of evidence there. So this was just some examples. So the bottom line is, if I go back to the whole grid, so you score 0 to 12 for genetic evidence, 0 to 6 for experimental evidence, come up with the total points, and that gives you either limited, moderate, or strong. If the publications have been out for at least 3 years and there's at least 2 publications, it's been replicated, and no contradictory evidence has arisen over that time, then you can move it to definitive. So that's generally how we've gone through this sort of semi-quantitative scoring process to document gene disease validity. And we have to do that really before you can start evaluating variants in a given gene. So maybe in a future time we can actually spend more time on the detail of that, but I think I'm going to stop there, allow any last questions, and then call it a day. Any other questions anybody has?